I can turn the lights down. Okay, one other announcement, or two other announcements actually. One of them I've got a reminder for later, but I'll do it now since I'm remembering. There is a question 13 on the evolution things. Did I ever give you question 13?
Yeah.
Actually, in print, in writing, or just orally. I actually wrote it down somewhere. I did. Okay, I couldn't find it, and I wasn't sure. Great. Okay, good. Okay. The other thing is that on these detailed reading assignments, under Introduction to the Nervous System, it says read V. Carter. It means read Carter. Page 48 to 49, 52 to 56, 40, and then again it says 48, 49. Just forget the second. I don't know what I was. I don't know what I was doing when I wrote it. But just cross out the second copy of the same pages and ignore it. I looked harder this morning, and it's not some other pages that I was referring to. I just wrote the same thing twice for some reason. I obviously revised this from something else where it didn't quite fully properly revised. Okay. So we are talking about introducing neuroanatomy, which we're going to need to know something about in order to talk about various things that I'll talk about, in order for Zidell to talk about things he's going to talk about. We had the brain, the spinal cord, and the brain. We have, if you look at a cross-section of the spinal cord, we did this last time. It looks like this. I actually can't quite remember where I ended. Did I talk about dorsal and ventral roots last time? No? Oh, okay. So if you cut the spinal cord in a cross-section, it looks like this, anywhere along it pretty much, from there to there. It's got gray matter in the middle, that is to say cell bodies, synapses, dendrites, so on and so forth. It's got white matter around the outside. Those are tracts of fibers running up to the brain and down from the brain, and also communicating from one part of the spinal cord to another. The gray matter has sort of an H shape. And we talk about these two parts up here as being the dorsal horn of the gray matter, and these two bumps down here being the two bottom parts of the H being the ventral horns of the gray matter. They're bilaterally symmetrical. The nervous system is organized in such a way that sensory nerves come into the dorsal horn, and motor neurons congregate in the ventral horn and send their axons out via the ventral root. Then later, not shown on this picture, those two things join, and you have nerves that have both sensory and motor nerve fibers in them. But I don't care that you know that in this course. That's everything, I guess, on that slide. Oh, okay. Did I give you question 13? And the answer is yes. Okay. Now, go up from the spinal cord, we go next to the brainstem. And on this slide of the brainstem, on the right, I have specific landmarks, and on the left, I have the divisions of the brain, the names of the divisions of the brain. So let's look at the landmarks on the right-hand side first. Out of your way. Maybe somewhere else. Okay, let's start down at the bottom. There's a bump there. So here's the top of the spinal cord. Here's the start of the brain. Like there's a bump there called the medulla oblongata, or often just the medulla. Everything on this slide you have to know, because these are things that people, not because it's important to know them, but because we will refer to things and assume that you know what we're talking about. So this is the medulla. Then up here on the dorsal surface of the brain, there is a big thing called the cerebellum. We'll talk a little bit more about some of these structures and what they do later. Others we won't have time to talk about. That's on the dorsal surface of the dorsal side of the brainstem. On the ventral side of the brainstem, we have this bump called the pons. Then go up here on the dorsal surface, we have two bumps called the superior and inferior colliculus. And this region as a whole, it's not on the slide. You may or may not have to know it. I'll come to that in a minute. This region here, which has a superior and inferior colliculus, is also called the tectum, which is a word meaning roof in Latin, I guess. Then if we go still farther up, we have a big ball here called the thalamus. And then below it, ventral to it, we have a mass of tissue called the hypothalamus. It's underneath thalamus. And then hanging off the bottom of the hypothalamus is a structure called the pituitary, which is a gland that secretes all sorts of stuff into the blood. That's it for the landmarks. Now, the divisions of the brain that these landmarks allow you to distinguish are starting again at the bottom. The part with the cerebellum, the pons, and the medulla all together is called the hindbrain. The part just here, going from the back, the part here, the bottom part of it is called the myelencephalon, and the part here that has the cerebellum and the pons is called the metencephalon. Okay? Then we go up to this division here, the part that's marked by the superior and inferior colliculus, and that's called the mesencephalon or midbrain. And then we go up here to the remaining part, and that's all called the diencephalon. And we'll be talking about some of these specific parts and what they do, but not others. The nerves of eyes, ears, so on and so forth, enter the hindbrain, and the motor, those are the sensory nerves, and motor nerves for the head, control the nose, the blinking of the eyes, and so on and so forth. Those exit from the hindbrain, part from here, various places along here in this region. Okay, now, if we go up a little bit farther, above the brainstem, we have the telencephalon, which includes the following things. It, first of all, as I said, just outside the top of the brainstem, the top of the brainstem, outside the thalamus and so on, there is a whole bunch of tissue sitting here on each side, and those are the basal ganglia, shown here in green. And they include the striatum, or sometimes more properly called the corpus striatum, and the amygdala, which I don't think I show. I don't. Most of it is a striatum. Most of the basal ganglia is a striatum. The amygdala is a little structure which is sort of over in here. I didn't show it on the picture. The striatum is basal ganglia, and then the striatum and the amygdala are the two parts of the basal ganglia. The striatum itself has a bunch of subparts, and these things, and I've listed them here, but you don't have to know them. If it's in small print, you don't have to know them. And I'll talk a little bit more about what you do and don't have to know in a moment. But these are structures called the striatum is made out of the caudate, the putamen, the globus pallidus, and some other stuff. The amygdala is a very old part of the emotional brain, which we're not talking about function right now, so that doesn't really matter very much. Then, as we go beyond that, we have the cortex, and the cortex has two parts. It has an old cortex and a new cortex. The cerebral cortex is the archicortex plus the neocortex. The archicortex evolved first, and what happens with the brain is things keep with the vertebrate brain is things keep getting added on top. keep getting added on rosters. So the first thing to get added on was some very old cortex, some so-called archaecortex called the hippocampus, and that's shown here. And then later there was a lot of other stuff added on, and that was the neocortex, and that's the rest of the stuff that I've not shown here. It's not colored here. And just at the sort of inside surface of the cortex is a particular kind of cortex called a cingulate cortex, which is sort of emotional cortex. And so those are where all those things are. So that's all everything that's not the brainstem, everything above the brainstem is the telencephalon. Now, if we go up to the neocortex itself, which is what we see when we look at the surface of the brain, what we see is a highly, it's got all sorts of mountains and valleys, which I've drawn here as smooth just because they make it easy to draw, but there's some particularly large valleys. These valleys are called fissures or sulci. They mean pretty much the same thing. And the most prominent ones are the central sulcus, valley, fissure, and the lateral fissure, lateral sulcus. This is the front end. This is the back end. Here's the cerebellum sticking down here in our pink brainstem. Now, using these fissures, these big fissures, as landmarks, one can divide the cortex up into different parts. It turns out that a neuroanatomist can also divide it up based upon other more detailed characteristics. But for our purposes, this will be sufficient. The part in front of the central sulcus here, all this stuff here, is called the frontal lobe of the cortex. If we draw an imaginary dividing line here from the end of the lateral fissure on back, and then from the top of the lateral fissure on down, we can then talk about the most caudal part of the cortex being the occipital lobe, and the part of below the fissure but in front of the occipital lobe is the temporal lobe. The region in here that's in between the central sulcus and this fissure and this dividing line is the parietal lobe. That's everything on there. So you got to know all that stuff. And here is a picture of what it actually looks like. I switched the brain around, so my diagram around so it would conform to this picture, because I couldn't turn this one around. So it's sort of not perfectly upright, so it's a little confusing. But so here is the temporal lobe. Here is the frontal lobe. Here is the lateral fissure, or the central sulcus, central fissure. Here is the lateral fissure. Back here is the occipital lobe, and so on. So this is what it actually looks like. So there are lots of other valleys and lots of other convolutions on the surface, which are not shown in this diagram.
Professor?
Yeah.
Does that mean that all the folds are in the same places in every human?
Pretty much,
Okay.
yes.
And
Not only in every human, but you can find the homologous folds in monkeys and so on, which is part of the reason. If you remember when I read some Darwin to you, he said all the primary sulci are in the same places, and and that's what he was talking about. Yeah.
And it's bilaterally symmetrical?
Yes,
Yeah.
bilaterally symmetrical. Yeah. Okay. Oh yes, right. Now, what do you and don't? Let's go back to this a moment. What do you and don't you have to know? There is a separate little sheet or document in the documents for the class on the website called something like extra brain pictures or Krasny brain pictures or something of that sort. I forgot exactly what it's entitled, which basically has two things on it. It's got a few extra pictures taken from other textbooks than Carter, because Carter is not really a systematic textbook. And it's got a table. And it will tell you on there, basically you pretty much have to know everything in the table, or everything in the table that I think it says on the sheet, it says you have to know things in bold but not things in not bold, or something like that. I forget exactly how it's phrased. That will tell you what you do and don't have to know. You have to know everything in that table. You not only have to know what those things are, you have to know kind of where they are. You should be able to point to all those things on one of these kinds of pictures. Okay?
Should we be expecting the picture to look more like this and less like the complex ones?
Ideally, any kind of picture.
Okay.
But I guess I use my pictures primarily because I'll probably, if I reproduce them on an exam, it's most likely I'll do that, but I don't always do it.
Okay.
Okay, so that's the end of the geography of the nervous system. Nothing interesting there. It's just stuff you got to know. Okay, now let's go on to the next part, which does have some interesting things in it, the functions of the various parts of the nervous system. That shouldn't be read there, that should be black, the bottom one. Okay. The way neuroanatomists or neurophysiologists, neuroscientists, have figured out what, in the earliest days, the way they figured out what the different parts of the brain did were by chopping bits off and seeing what was left, seeing what the animals with what they had left could do and could not do. And there are certain classical cuts or removals that they made. I'm not going to talk about all of them, but I'm going to talk about some of them. So the first one they made was to make a cut at the top of the spinal cord, or sometimes in the middle of the spinal cord, and study the parts of the body that were controlled by the part below the cut. In these diagrams, I'm going to put in red the highest area still connected to the limbs or to the body, and the part that we're studying, the behavior of. So like you make a cut here, don't remove the tissue, all the stuff up here is still connected to the head and so on and so forth, but we're going to ignore what the head is doing. We're only going to look at the parts below this cut of spinal cord are connected to the arms, the legs, and so on and so forth. And in studying this animal, we would only pay attention to what parts innervated by the part below the cut we're doing. Because what we want to know is what is this red part capable, what kinds of behavior is it able to produce, what are its capabilities. This kind of animal is called, it's prepared in this way, it's called a spinal animal. It's named for the highest part that works, or that's being studied. This very famous neuroscientist by the name of Sir Charles Sherrington got the Nobel Prize in the end of the 1800s for his work on this. He learned an awful lot about how the nervous system works from studying the behavior of spinal animals. And here are some of their characteristics. First of all, you might think, well, if you've only got a spinal cord and no brain, you would just be a puddle on the, fall down and be a little thing lying on the ground with no movement. That's not true. There's a lot more going on in the spinal cord. The spinal cord can do a lot more than that, though there's an awful lot it cannot do. So first of all, these guys, in fact, cannot stand on their own. That's mainly because there's several reasons, but one of them is the organs of balance are above this point, so they don't have any organs of balance. And there are some other reasons too. But you can put them in a hammock. So their legs are down. Usually you study a quadrupedal animal, usually a cat or a dog in Sherrington's case. And so their legs can be touching the ground, and so they're kind of upright. So he studied animals like that a lot, so they can't stand. Moreover, if you put them in a hammock like that, they never do anything spontaneously. They just sit there. But they do respond to certain kinds of stimuli. So, for example, ignore that one for a moment. We'll come back to it. For example, if you take an animal like this and you pinch its toe, withdraw. If you pinch its foreleg, it'll withdraw. These withdrawal reactions involve flexions in almost every joint. So you're sort of trying to scrunch up and get it away as much as you can. And that's what the animal's trying to do. And so, because it involves a flexion at every joint, it's called a flexion reflex. But there is wiring in the spinal cord that ensures that whenever the animal makes a flexion motion, which contracts the muscles that cause flexion, the muscles that cause extension get relaxed. So you have, and that's due to some sort of inhibitory process, which we'll talk about the nature of inhibition later on. But so in the flexion reflex, you have flexion and then all the antagonistic muscles that are antagonistic to the flexion the motor neurons get inhibited and the muscles relax. So it's a coordinated movement. If that didn't happen, you tried to contract both the extensors and the flexors at the same time, you would tear a tendon or do some damage. So that's built, and that happens due to circuitry that's built into the spinal cord. Another thing that happens is if you poke the animal's right toe, it lifts this leg. But of course, in real life, if it lifted this leg, its full weight would then have to be borne by the other leg. And so what happens is at the same time that it lifts this leg, it increases the extensor thrust on its other leg so as to take up the extra weight. That's all automatic. That just happens due to circuitry, neural circuitry within the spinal cord. These actions that I'm just telling you, that I just told you about, are all kind of monophasic. That's how you do something and there's a single response. But a very important kind of thing that all animals do, of course, is things that continue on their own in time, like walking, flying, so on and so forth, and breathing. These things involve self-generated activities are self-generated by the nervous system. They're not reactions to any external events. I mean, they may be a reaction to an external event, but the continuation of the action isn't usually due to an external event. In other words, once you start walking, you might walk because somebody gave you a kick, but once you start, you will continue because it's generated by your nervous system. The kick is gone. And these kinds of repetitive actions can also be produced by the spinal cord. It doesn't need the brain. The example that Sherrington studied the most was the scratch reflex. So if you take a dog and tickle it appropriately, it will go, like that. Even if that's mediated entirely by the spinal cord. So the spinal cord can produce these repetitive, ongoing repetitive rhythmic activities. With this animal in a hammock, if you poke the toes in the right way and so on, you can actually get a step or two, just mediated only by the spinal cord. And, of course, there are all sorts of tracts of fibers running up and down the spinal cord. In an intact animal, there are all sorts of tracts of fibers running up and down between the spinal cord and the rest of the brain. If you take a suitable, and this was discovered after Sherrington, it was discovered later on, if you take a suitable, if you activate just at random nerve fibers, may produce nerve impulses in nerve fibers that are descending down into the spinal cord from above and get the right ones. Even though it's totally random activity, you get the right descending axons, this animal will actually start walking, even though he's only got a spinal cord. And if you put him, you can actually, depending upon how strong you stimulate him, you can get him all the gaits. You can get, in a four-legged animal, you can get trotting, walking, galloping, all these coordinated gaits, all mediated by the spinal cord. So all the circuitry, all that kind of stuff is in the spinal cord. Yes.
So this is a dog with no head.
Right. Well, he's got a head, but you're studying the part of him below the cup, and he's still got a head. What the head is doing, what it's doing. We're not paying any attention to it because there's a cut here. So if you stimulate down here, the rest of the brain above that point doesn't have any idea what's happening.
Okay.
And the front end of the dog is doing what he's doing.
Okay.
He may be licking you or eating his dinner or whatever. Yeah. We're just paying attention to what the part that's controlled by the part below the cup.
Okay.
We don't actually remove all this tissue. If we didn't kill the animal.
It just means we only stimulate the part below that.
We only stimulate the part below. We only pay attention to what the part below is doing.
Okay.
Now, these responses that occur in response to stimulation in a spinal animal occur, but they are automatic. They don't involve the mediation of consciousness. Things that don't require the mediation of consciousness we call reflexes. So these are all reflex responses. And one can show that they don't involve consciousness in several different ways. A good example, the one that's been studied a lot, is the flexion reflex. So you pinch or stick a pin or whatever in the toe and get a retraction like that. If you do that in an intact animal or an intact person, what you find is that, and time, exactly when it starts to hurt, and when the animal starts to move, or in the person, when the person reports pain or whatever. And you can tell when it starts to hurt the animal by looking at emotional reactions and so on and so forth. You find that the flexion reflex occurs before the animal seems to know anything at all has happened. There's no sign. The flexion reflex occurs before the rest of the brain probably even finds out that you stuck the pin in its toe. So these things are, that's one kind of evidence that these actions do not require the mediation of consciousness. Another way is the tracts that carry information about the pinprick to the toe are the so-called spinal thalamic tracts, which are ventral spinal thalamic tracts, which are shown here in pink. And there are some humans that have, due to car accidents or gunshot wounds or whatever, have had these tracts severed, at least on one side. And so the brain doesn't get this information above the point of severance. So if you stimulate the animal on, say you stimulate its foot, so say such a person walking along, this has actually happened. Neurologists have described this. Person who has this severance somewhere up here in their spinal cord of the ventral spinal thalamic tract steps on a nail, a pure nail by accident. And the description that I sometimes read, I'll just tell you what the patient did. The neurologist was describing is the patient said, I stepped on this apparently, and I lifted my leg and I looked down to see why my leg had contracted. No sensation, no feeling of the pain, not even pain, no conscious sense of any stimulus at all. Just the spinal cord did it. The brain didn't find out about it. So that's why we call this a reflex action. It does not require mediation of consciousness. Okay, so that's it for the spinal cord. Now, I always ask questions about these things, and the ones I'm going to show you on exams, and I actually would like you to know them. I always forget to say that, and people miss a lot of them. I actually want you to know them. Yes?
Does that mean that only the pain part,
I'm sorry, what?
Does that mean that only the pain part will transfer the feeling of pain to the brain?
I'm sorry, say that once more.
Does it mean that only the pain parts will transfer the feeling of pain to the brain?
Wait a minute. So these tracts here carry the information that allows the brain to feel pain.
Only this part.
Yes. Yeah, it's not going up everywhere. Different tracts going up carry different kinds of information. Yeah. Whoops, sorry. Okay. Now, the next kind of preparation going up we're going to talk about is one where the midbrain is the highest functional part that's working. And this is produced by making a cut above midbrain. And again, the rest of the brain is still there, but we're just not paying attention to it. So these guys still have very little, if any, spontaneous behavior. In response to stimulation, they now can stand, walk, and climb. So they have a fancier set of responses to stimulation, partly because they have organs of balance and for some other reasons too. If they can do coordinated activities like crouching and defecating in the case of a dog, if you put food in their mouth, they'll swallow it in a coordinated way. If the dog, for example, and you make it wet, it'll shake the way dogs do to shake off the water. Rats will groom, and so on. But it doesn't do any of these things in a meaningful, integrated way in the service of goals. It certainly doesn't ever search for food if it's hungry. It doesn't. I mean, it just basically, it's still pretty much of a, in quotes, a vegetable in the sense that it never does anything on its own. But it's got fancier behavior than just a spinal animal. Yeah. If we go still farther up, the next thing we do is look at an animal that, the animal that I was just talking about was called a midbrain, or mesencephalic, because the midbrain is the highest part that is mediating the behavior we were studying. If we go still farther up and ask what an animal that has its basal ganglia working we get still another picture. Now, the way these animals are actually produced is a little more complicated. What's done is we don't want the hypothalamus in this preparation, but we can't remove the hypothalamus because it'll kill the animal. So what we do instead is we sever the hypothalamus from the rest of the brain, but the hypothalamus is still there and able to secrete the various hormones and things that it normally secretes into the blood system. If you don't do that, the animal doesn't make it. So it's still there, but it's not neurally connected to the rest of the brain. So we remove the hypothalamus. Sorry, I didn't say that quite right. We cut this, and so the pituitary is still connected to the hypothalamus and still can secrete things. But the hypothalamus is not connected to the rest of the brain. And we make a cut up here to get rid of the connection to the neocortex. The thalamus is still there. We don't get rid of the thalamus, but it turns out that what the thalamus does, it's a communication pathway between the lower parts of the brain and the neocortex. So with the neocortex there, the thalamus, though still there, is redundant. So it's still there, but it's not doing anything important. That may be a slight oversimplification, but that's at least what people thought at the time the experiments were done. And for our purposes, it's good enough. Okay, so we're studying this animal that has its basal ganglia. It does not have or use its thalamus. It does not have its neocortex. In this case, we actually remove the neocortex. And it's not going to be using, for the purposes of the behaviors we're going to be studying, it's not going to be using the hypothalamus. So the hypothalamus is still there, connected up to the secretory organ. Connected up to the pituitary, which in turn it can still secrete. Okay, so what does it do? Still responds mainly to stimuli. Still no spontaneous behavior. Behavior as a whole is qualitatively similar to the midbrain animal, but somewhat richer. This animal will chew or lap and swallow food if it's deprived, but if you put food in its mouth and it's not deprived, it'll spit it out. It won't ever look for food or water, so although it can apparently tell the difference whether it's hungry or thirsty, in the sense that, like, with the food, it spits it out if it's not hungry. It doesn't ever look for food if it's hungry. It just basically is kind of passive. And if you try to force food on it when it's not hungry, a normal animal, if you try to force food on it when it's not hungry, will make a fuss. These guys don't ever make a fuss about anything. They lack normal motivation and normal motivation and normal emotional reactions. These basal ganglia animals. Okay, so this picture is kind of similar to a little richer than what we saw with the midbrain animal, but not a lot so. But if we now remove that cut between the, if we no longer sever the connections of the hypothalamus to the rest of the brain, but everything else is still the same. The cortex is still gone. We have what is sometimes called a hypothalamic animal, but it's got the basal ganglia plus the connected hypothalamus. Sorry, it's more often called a decorticate animal, because that's what's really the main thing you've actually done is you remove the cortex. The whole picture changes. Now we have all sorts of spontaneous behavior. The animal is hungry, it searches for food. If it's thirsty, it searches for water. It will learn to do things, simple things, to get food. If you try to feed it when it's sated, it'll fight you off. If you annoy it, it will attack. In rats, it can even rear a mother rat who doesn't have any cortex to still rear her pups. So it's got a lot of behavior. Now it's got two different kinds of learning, which now is a good time for me to mention. There are two major kinds of learning that psychologists study and that we ourselves do like. One of these is like Pavlov's dogs, who most of you have probably heard about. If you present food to a normal dog, it salivates like mad. If you ding a bell to a normal dog, who hasn't been trained, they could care less. However, if you systematically, on many occasions, present ding the bell, give him food. Ding the bell, give him food. Over and over and over again enough times, the animal learns an association between the bell and the food. And then if you ding the bell, he salivates and shows other signs of expecting food. That is called classical conditioning or Pavlovian conditioning. Pavlov being the guy who discovered this kind of learning. Pavlov had already gotten a Nobel Prize for the study of digestion. He didn't get another one, but he started the systematic study of learning. The other kind of learning that's extensively studied, and that we do, is called instrumental learning. It's learning to do things in order to get things, or in order to get away from things, to not get things. And it's instrumental in the sense that you do something in order to. The doing is instrumental in achieving some end. And an awful lot of the learning we do is like that. In the laboratory, we study, the classical study of this is rats studied by a psychologist named Skinner, who learned to press a lever in order to get little pellets of food, or he studied pigeons who learned to pet the key, little lighted key in order to get bits of grain and so on and so forth. Rats will learn to run a maze and learn the right, which is the side the food is on and on. That's all instrumental conditioning, instrumental learning. Sometimes called operant conditioning or operant learning, but instrumental learning is probably a better word for it. So anyway, these guys without a cortex can do that perfectly well. Both those kinds of learning, perfectly well. They can't distinguish many kinds of stimuli that an intact animal can. So their sensory capacities or their perceptual capacities are diminished. But if you use simple stimuli, and they can learn these things. And also, if you ask them to perform a response which requires whole body movements, walking, flailing about, whatever, gross movements of limbs, that's swell. If you ask them to do something fiddly, they can't. To do fiddly things, fine manual skills, they need their cortex. So they need their cortex for certain kinds of important sensory processing and motor production, but the basic abilities to learn and perform and react to stimuli are all there, even without the cortex. Okay, now we add on the cortex. What does the cortex do? The cortex is divided into these divisions that we talked about, the frontal, parietal, temporal, and so on. And it turns out that, as Zeidel will talk about in extensio, and I will talk about too to some extent, there is tremendous separation of function, sort of modularity in the nervous system, with different parts of the nervous system doing different jobs. And that is particularly apparent in the cortex, and it was first discovered, I think, mostly in the cortex. It turns out that, and so this picture shows what some of the different parts of the cortex are important for. So visual perception, at least the earliest parts of visual perception are back here. Some parts of visual perception are here in the temporal lobe too, but the earliest, the most elementary aspects of visual, analyzing visual scenes are back in the occipital, back part of the occipital lobes. Higher order visual perception is here, in the so-called inferotemporal cortex, the bottom part of the cortex. And there's also appreciation of where you are in space, the shape of your body in space, is here in the parietal lobes. The perception of tactile stimuli, somatosensory stimuli, tactile, touch, heat, cold, things like pain, are here just behind the central sulcus here, the so-called post-central gyrus. That post-central gyrus is in small type, but it shouldn't be. You do have to know that. Post-central gyrus. In front of the central sulcus, we have the control of movement. We have, and especially of fine movements. And in fact, the precentral gyrus here, and I'm sure there are pictures of this in Carter, is sort of a map of the human body. where if you stimulate one part, you get movements of the fingers. Stimulate a different place, you get movements of the toes. Stimulate a different part, you get movements of the nose, etc., etc., etc. And in fact, there are pictures of the so-called homunculus, of a little man standing upside down, where his head is down here and his feet are up there. And if you stimulate, or if you make neurons in this region here fire, you'll get movement of muscles that control things in the head and face. And if you do it here, it'll be movements of the feet and so on. So there's detailed control of the body by nerve cells in front of the central sulcus. This is called the pre-central gyrus. Here on the temporal lobe, there's auditory perception. And then the more front parts of the frontal cortex are various kinds of planning activities. Immediately in front of these sort of primary motor areas in the precentral gyrus, there are regions that are involved in making motor plans. Plans, for example, if you have a monkey with, it's got a plexiglass screen, the hole in the screen, and there's food not immediately behind the hole, but off to the side. The monkey can see it, but it can't actually get to it by just going like that. It's got to go around the hole. Organizing that movement of putting your hand through and going around the hole, that's organized in this motor planning area in the middle part of the frontal cortex. And then in the very front part of the frontal cortex, the so-called prefrontal cortex, is general planning ability. Planning your day's activities. People talk about this being the executive controller of the brain. It makes, yes?
I'm sure it's to know all the functions of it.
Yeah. And I guess that's everything there. And it said, I think I already said this. Cortex is needed for learning of complex discrimination, visual, auditory, whatever, and for fine motor skills, but not for simple discriminations, learning one thing to do one thing to one stimulus, another thing to another stimulus, so long as the stimuli are simple. Cortex isn't needed for that. Nor is the cortex needed for any kind of learning involving gross movements. It's only fine movements that absolutely requires the cortex. Now, of course, learning to do things that involve complex activities that involve planning or motor planning, we do need the cortex. Okay. So that is, if we go back to our overall, come on. This wasn't worth it. Okay. So we've now done all this, and now we're going to talk about how these neurons themselves work. I've given you already a little introduction to the anatomy of the neurons, but how do they work? Okay, now, so how neurons work. There are a number of things we're going to talk about here, and I'm not sure I will make clear exactly when we're talking about which, but you can look at this list after we've gone through it and make sure you know what each of these has to do with. If you know something about each of these things. There's a demonstration here that I think we probably won't have time to do, so I'll come to it later if we do have time. Okay, so here's our nerve cell again. Same picture that we had before, with the cell body and the dendrites and the axons, the axon terminals, excitatory inputs, inhibitory inputs. If you take a nerve cell like that, when it's at rest, it's not doing anything, just sitting there, and you put, well, sorry, let me back up. The signaling and the information processing that nerve cells do is largely an electrical event. It's electrical signals that the nerve cell uses to communicate. These are electrical signals that are based on chemical events, but they are electrical signals. And so the electrical properties of the nerve cell are very important. Now, if you take a resting nerve cell and take a voltmeter and put one poles, voltmeter outside in the body fluids that are surrounding this neuron. And the other side you attach to a thin electrode, a wire that's that can be made in a very fine way, so you're going to be able to get it inside the nerve cell when you want it. If you just put this, so you got one pole of the voltmeter here and the other connection outside the nerve cell, you won't measure any voltage difference. There'll be measuring between one place and another in the body fluids, and there's no voltage difference between them. But if you now push this, and that's so if you measure the voltage on the scale minus, well, on the scale, a positive voltage, a negative voltage, this is 0. If now you poke this electrode through the cell membrane, so it's on the inside of the cell, but this other electrode is still outside the cell, so you're now measuring the voltage between the inside of the cell and the outside of the cell, you do get a voltage. And that's a voltage that is inside negative. The inside of the cell is negative relative to the outside of the cell. And it's negative by approximately 100, about a tenth of a volt. Did I say that right? 100, yes, it's about a tenth of a volt. It's 100 millivolts. A millivolt is a thousandth of a volt, and it's about approximately 100 millivolts negative. The inside is approximately 100 millivolts negative to the outside of the cell. That voltage is called the resting voltage. It's there all the time, unless something's changing it. It's the standing resting situation, the resting voltage. But physiologists don't use the term resting voltage. In physics, another synonym for voltage is potential. And this is usually referred to normally not as the resting voltage, but as the resting potential. Means the same thing, but nobody ever says resting voltage. They always say resting potential, so you have to know that. So there's this resting potential. So on a graph like this, where we have plus, if you look at the inside of the cell relative to the outside of the cell, which is the conventional way that physiologists talk about the electrical properties of a nerve cell, if you graph the, or if you make put on a graph the voltage of the inside relative to the outside, there's a 0 voltage when this electrode is outside the cell. When it pops into the interior, the inside goes 100 millivolts, approximately, a little less than that, negative to the outside. It actually really goes somewhere between 60 and 90 millivolts negative when the electrode goes into the cell. Okay. Let's continue this next time.